Our next speaker is Heike Sichtig. Dr. Sichtig is a team lead and principal investigator and regulatory scientist in FDA's Office of In Vitro Diagnostics and Radiological Health in the Division of Microbiology Devices. And Dr. Sichtig will be talking about the regulatory perspective for infectious disease diagnostics and the FDA-ARGOS database. So thank you.
Thank you. To just use this to advance. Okay. Hello. Welcome this afternoon. I'm Heike Sichtig. I work at the FDA in the Center for Devices. And I've been told that I should always start out my presentations with a joke. But my humor is German, so you probably won't get it. Also, I've been told that I have an accent, but I don't really hear that. So I'll be giving you a talk, and I think it's the last one today, which is on regulation of these type of devices, and also give you an overview of the FDA-ARGOS database. Disclaimer that we all have to show is opinions are my own, really, and the policy on these type of devices using next-generation sequencing is still developing, and any thoughts presented are preliminary. So what I wanted to do in the beginning is walk you through how we at FDA go about regulating these devices. We look at risk first, and everything is sort of binned into 3 different risks. Is it a high risk? Is it low risk? Is it a medium risk? And at the microbiology division, typically what we see is a class 2 device, because that's where they get classified, which would then require only special controls and what we call a 510K submission, or if the technology is not out there, it's a de novo submission. However, if there are analytes on the device that would put it into class 3, then it might require a pre-market approval, but not to worry, it's also not that big of a deal. It doesn't take that long, as it used to be. So our current evaluation of diagnostic devices is sort of outlined on this slide, and it's the general concept on how we look at things. The device comes in, just assume it's a black box, whatever the technology is, we don't know what it actually does. And then there has to be a reference method to assess the performance. So we do a 2x2 performance, and we look how the device performs. And it starts with a sample, which could be a bacteria, a virus, or fungi, that goes into the black box device and also into the reference method. Now you can see with next generation sequencing, especially for metagenomics, where you could potentially identify many microbes simultaneously, this is a big problem. Because now each possible organism would need confirmation by this reference method, and it becomes extremely burdensome. We've always been evaluating the whole entire system, starting from sample prep all the way down to the database, including pre-analytical steps, amplification, the sequence detection reads, and also the database algorithm. What we've done also is assemble together a multiplex and microbial sequencing in vitro diagnostic team, especially to address these issues, where we expect that companies are going to come in and want to have claims for detection of multiple pathogens simultaneously from a single clinical specimen. And this is not just for really the devices that we're talking about, but it could be useful for emergency use as well. So if there's an outbreak or something is happening that we're not prepared for, NGS could potentially be used. So we've been at FDA thinking about potential pathways on how to move it through the regulatory pathway more quickly. We're all aware there's lots and lots of publications already. People use it in their hospitals, in their labs. And the only really the burden right now is to figure out a way on how can we move this through regulation, and how can we make codes, and how can possibly hospitals get reimbursed for using this technology. I think this is one of the biggest issues, especially for infectious diseases. We published a discussion paper on our current thinking. That was about 2 years ago, where we also had a workshop. I don't know if any of you attended, but there some scientific and clinical community leaders expressed that oversight of these type of devices is needed, and if we can provide some guidance. And what we've done is also looking at one side is the human sequencing, which has very different issues versus infectious diseases, where NGS is used, because here we are really looking into an absolute need for an immediate and actionable result. So for cancer screening, it's okay to go over a couple of days or weeks or even months. For microbial sequencing, that's not the case. We need to have a very short turnaround time. Also here, we're looking at a broad range of specimen types. We are looking at a large diversity. We're looking at microbes moving around spatiotemporally. So it's not the same problem talking about human sequencing versus microbial sequencing. And we tried to address that in the discussion paper, and we've also been working on a guidance document that hopefully should publish soon. We also worked on or have been initiating an FDA-ARGOS database effort where we like to provide the community with regulatory grade reference genomes. And I will go into more detail in the following slides. But first, I'd like to give you an overview of what is, if anybody's interested, what a submission would look like and what the proposed information would be. As part of that submission, it would be a assay description, then you would also have a validation, if anybody's familiar with that, then analytical performance, instrumentation software, and clinical evaluation. And here's where the difference is. Most of it is similar to the multiplex guidance that we provided, but for metagenomics, there's differences that need to be addressed. And also what we're trying to do is provide the community with reference genomes via FDA-ARGOS that could potentially be used as an in silico comparator. This is so for NGS, because it's a new technology, most likely whatever we're going to see first will be what we call a de novo submission. And again, this is based on a risk-based pathway for clearance. And that means that appropriate devices are not really available, so there's no predicate that can do this kind of technology. And also the characteristics would raise new questions of safety and effectiveness. You can always use the pre-submission process, and we can share whatever information we have currently. There's a list of recent de novo clearances. I don't know if you are aware, but you can go, I know it's like very difficult going onto the FDA website and looking for information. I use Google. But there are databases that you can search, and there's a 510K database, there's a PMA database, and they also recently put a de novo database, and you can look through them and find information like the decision summary, the performance characteristics. All this information is public. Coming back to next generation sequencing, it is easier to sort of think of it in two buckets. One would be more like the PCR approaches, the multiplex PCR approaches, versus on the other side we have metagenomics sequencing. For metagenomics, we don't really have any a priori knowledge of the sequence targets, and we can generally identify all targets in the same sample. So this raises new questions, and our current challenge really is to move these NGS-based diagnostics into the microbiology lab. What we heard before as well, it's for Mycobacterium tuberculosis is also applicable to any other microbes. And what we really need is a robust, standardized, and high-quality microbial sequence database where we have representative samples, metadata, high-quality raw sequences, assemblies, annotations, and all this should be in the public domain. What we put in for funding to get this effort started back in 2012, 2013. And ever since, we've been working on adding about 2,000 high-quality, MCM stands for medical countermeasures, it's biothreat organisms, and clinically relevant pathogen sequences. We would like to add those into the public domain. And we established this expanding publicly available database, which is hosted at NCBI, so we didn't create our own database somewhere because there's already a great structure available at NCBI. So we integrated into there and created a bioproject. It's called FDA-ARGOS, and it meets regulatory grade quality criteria, which I will go over in a further slide. And it is really critical for developers to validate these candidate high throughput sequencing based in vitro diagnostic assays. So what we envision is when the diagnostic devices come in for clearance approval, there will be claims on detection of these organisms. And then what we hope to accomplish is having an in silico comparator available through the database, because now it's not just we're getting sent NCBI accession IDs and we have no idea how this sequence was generated, but we know exactly how it was generated because we put it there. So we have the raw reads available, everybody can go back and revalidate, we have the assemblies available, and we have the traceback of the metadata as well. We are collaborating with the DoD, with the NCBI, and also with the University of Maryland Institute, who does all the sequencing for us at the moment. And we try to include geographically diverse isolates from different agencies. Like I said, around 2,000 will be sequenced, and we also like to include antimicrobial resistance isolates. It's not a mandate of the database, but it's a nice feature. So with a lot of the isolates that we collect, especially from the Children's Hospital, we have antimicrobial resistance data. So we have the genotype, phenotype information as well with these isolates, but this is not a requirement. I've listed the bioproject ID if somebody is interested. It's available, it's up right now. You can download any of the information that is public right now. We are going through sequencing of the genomes in batches of 100. So we are working on some more press that we can notify people when the next 100 genomes are released. Right now we have about 2 batches available, and we're working on the 3rd batch to be released very quickly, and then it will be ongoing. As I said, we are collaborating with many agency clinical labs and repositories. If you're interested, just let us know. Right now, we still have funding available to include isolates. It's a good time if you want to have it sent to our pipeline. And what we also like to do is qualify genomes. So there's all this great information already out in the public. But what is really helpful for us right now is have initial genomes, sort of like 5 sequenced with our pipeline, and then we can use that and qualify 1,000 other strains that are already available in the public. The sequencing center is the University of Maryland, the Genome Resource Center, and we are applying a hybrid approach using PacBio and Illumina data. And we deposit the raw reads, assemblies, and annotation at NCBI. And we're working on creating an FDA interface, because I find it also very difficult to navigate through NCBI, where you can go to a bioproject and you can download all the data, but what if you're looking for a specific one? It's sort of difficult to go into data that is hosted in biosample, data that's hosted in assembly, data is in all kind of different databases. So what I would like to do is have one website where we can access this information more easily or more simply. It's like you're interested in this organism, you want to download the raw data, or you want to just download the assembly, and that should be easily accessible. This is an overview of the project approach. Like I said, we apply or we use Illumina sequencing at about 300x coverage, and we use PacBio sequencing at about 100x coverage. And then we also piloted Ebola genomes, and we're interested in including more viral genomes. And that typically we use for shorter genomes just Illumina, and we do shotgun sequencing, amplicon, and race, and that provides more than enough coverage. We also do some of the data with PacBio, but there's not that much added information at the moment, so sometimes we do both approaches and sometimes just Illumina. Then we do the assembly and annotations. So every time a genome is produced, we run PacBio only, Illumina only, and the hybrid PacBio Illumina assembly. And like per organism, maybe we get 16 or more assemblies, and then a bioinformatics expert sort of goes through it and chooses which one is the best. And then we also have regular update calls where we look at the data, and FDA then says, like, this is fine to submit and move forward to the public repository. So in a way, it's really nice to have this feature, because what we've been able to do is, from the submitter, get the ID of the organism, do a de novo assembly with this pipeline, where we're getting a second ID, and then a third ID we get out of NCBI, where they provide also services where they put it into their phylogenetic analysis and can tell us, hey, this has been renamed. What should we do with this? So it's a really nice setup. Some of the data that get generated from PacBio, the base modification data, we're still working on how to make that available publicly, but we have it. So we hope that we are addressing part of the public health need of making a robust, standardized, high-quality microbial comparator sequence database. And I just wanted to also mention that we're working on reference materials, which is very, very important for these devices. You imagine using the database as an in silico comparator. How do you test the entire pipeline? From sample all the way down to diagnostic results. So something like a reference material might be applicable. And we've discussed this at length. At first, we have worked with NIST to create 4 organisms that are being made available, I believe, as a 4-pack. And then we recently developed another workshop where we discussed making standards available for mixed samples. And that is ongoing, and if anybody is interested, just let me know and I can add you to the list. But we're starting to work with NIST on creating these as well. And that should be a little bit more flexible, because typically it takes a very long time to create a reference material. We don't have that many years to create a reference material. We kind of need it now. So we are discussing how can we make this available in a short turnaround time. So this is just a summary of the different activities that we've been involved in. One is the FDA-ARGOS database, which takes up a big chunk of time. We've also been working on the guidance document, and I'm not allowed to say any times when it publishes, but soon. Then we have a interagency working group looking at NGS feasibility, which has been very helpful and informative on looking at some of these studies, getting into detail. And we have a collaboration with NIST on microbial reference materials. And we're supporting them definitely to make this available. And you can always use the pre-submission process for any questions. And that's the acknowledgement slide. There's so many people involved. It's very difficult to fit all these names on one slide. Thank you.